Hello, welcome back. Here's our mini lesson on translation. So kind of the final step of our central dogma. So we've covered transcription. Now we're going to do translation, converting that language of RNA nucleotide sequences into amino acid sequences. Before we get into jumping right into the process, I wanted to take a little bit of time to talk about tRNAs, those transfer RNAs. So because they're an RNA, they are transcribed from a gene in DNA. So it's a gene that codes for a particular tRNA. But then instead of being translated like mRNA does, it just is folds up into this functional RNA. And it has lots, depending on your resource, it's got some funny shapes to it. So I'm just going to draw something that kind of looks like that. Okay. Sometimes it looks more like a T. Your diagram that we're going to go into makes it look like a U. But there's two really important parts of a tRNA molecule. So we have down here kind of the exposed end of the tRNA molecule. This is where we're going to find what we call the anticodon. Okay. All the anticodon is, it's a complement to the codon. So it's just going to be some exposed nucleotides that will have a complementary sequence to the codon for a particular amino acid. And then that amino acid is going to be hooked up here to the top end. So for example, if we have a piece of mRNA coming down here, and I'm just going to use the codon that I know because that's the only one I know by heart, is AUG. Okay, so let's say this is the codon, right? So this is the codon on the piece of mRNA. Okay. So now I'm going to put, because I know which amino acid this codes for, it codes for methionine because it's a start codon. I know that. So what would the anticodon be? So the anticodon is just the complement to the codon. So in RNA, A goes with U, U goes with A, and G goes with C. Okay. So for any time the ribosome reads the AUG codon, the only tRNA that will come in with its anticodon, this complementary lettering, UAC, it's the only one that's going to come in. And the tRNA that has the anticodon of UAC will only always have the amino acid methionine. So that guarantees that whatever the codon is, the anticodon is going to be bringing the correct amino acid. So for all of those 64 boxes, with the exception of the stop codons, those 3 stop codons, every single one of those amino acids, or the codons that code for an amino acid, there's going to be a corresponding tRNA. So there's going to be a tRNA for all of the codons for leucine and isoleucine and alanine and phenylalanine and glycine and valine and all of those amino acids. You're going to have a corresponding tRNA that's going to recognize those codons in the mRNA. Now, these are reusable. So when this tRNA comes in and brings the correct amino acid, it's going to drop off the amino acid and then go back into the cytoplasm and get recharged to have the correct amino acid. So once it drops off this methionine, my empty tRNA for methionine, that UAG will go back into the cytoplasm. There's an enzyme called the aminoacyl tRNA synthetase, that's a mouthful, will find that substrate, the tRNA, and using energy, ATP, hook a new methionine onto that UAC methionine tRNA. And so it'll be ready to bring in another methionine if an AUG shows up in the mRNA sequence. So I just wanted to mention that tRNAs, transfer RNAs, their job is to bring the correct amino acid from the cytoplasm to the ribosome for the process of translation. Okay, let's get to it. So just like we had in transcription, there's 3 stages: initiation, elongation, and termination. So we're going to take a look at the initiation first. Now, if you remember, ribosomes are kind of a 2-part organelle. We've got the small subunit and the large subunit. And the mRNA is kind of sandwiched between the 2 subunits. the way that this starts, right? So we have our 5' cap and our 3' poly A tail. So this is our mature mRNA. It comes in and we're going to have the AUG is the start codon. So this small ribosomal subunit comes along and scans that mRNA and scans it with that tRNA. Here's your tRNA, right? They're making it look like a U, but it's just kind of schematic here. So it's going to have that tRNA with the methionine already hooked to it, kind of in association with that small ribosomal subunit. It kind of scans along until it finds that AUG start codon. And so that's going to be this position right here. So once that happens, once we've had the methionine loaded tRNA recognizing the start codon, the AUG, this is when we're going to get the large subunit to come in and sandwich in on that, because that's where we're going to start. Now the large subunit has 3 spots in it. We have what's called the E site, the P site, and the A site. Okay. The P site, where the methionine loaded tRNA is going to start, this is called the peptidyl site. This is where the peptide bonds are going to be made. Remember, peptide bonds hold your individual amino acids together. The A site stands for the amino acetyl site, because it's the amino acyl tRNA that's coming in. But I'd just like to call it the arrival site, because that's where the next tRNA is going to arrive. So that kind of makes sense. And the E site is what's called the exit. So once the tRNA drops off its amino acid, it makes a peptide bond with the previous amino acid that was dropped off. It kind of shifts down into the E site, and then that used tRNA will go back into the cytoplasm and get recharged with its proper amino acid. So this is what we call the initiation complex. We are ready to start. We've got our large subunit and our small subunit sandwiched over the mRNA. The amino acid, or the tRNA that's bound to the methionine amino acid, is in the P site, and the A site is open, ready to read this next. Oops, let's get a more vibrant color here. Is ready to read this next codon. Okay, so that's going to get us into the elongation. So initiation, we start with a methionine tRNA in the P site. Okay. So in this particular case, they've already added 3 amino acids, right? So we're looking into the 4th codon coming in. So here we have our open A site, and there is our codon, red, red, green. I don't know what those letters are representing. But if we're looking for the anticodon, here's the anticodon. It's going to be complementary, so green, green, red. That kind of makes sense. Here is its proper amino acid that's hooked to that tRNA. It's going to come in and bind to the A site. Okay. We burn a little bit of energy. It's using GTP, which is an equivalent of ATP. And so what that does is it kind of sets up this amino acid right next to this amino acid. And then the large subunit has enzymatic properties. Ribosomes are a combination of rRNA, which is kind of the structural piece, and protein. And proteins are enzymes, so there's an enzymatic reaction that's going to hook that 3rd amino acid to the 4th amino acid. And so that's our new peptide bond. Yay! So we've just added our 3 hanging off of that P site tRNA to that 4th amino acid that's coming in, that's hooked to that A site tRNA. And then the whole thing shifts, burns a little bit of GTP energy, things shift down. Now, what was our A site tRNA is now in the P site, and what was our P site tRNA now goes to the E site, and it's just ejected out, exit in the E site, and it's going to go and get loaded up with another of the proper amino acid, and we are ready to bring in the next tRNA that's going to have the complement to that particular codon. So that's purple, green, green. So then it would be, I don't know what the complement to purple is, blue, red, red, if that's the anticodon. And it would bring that in, and it just continues, just like chugging down the line. It's going to read each codon. The tRNA is going to come in, bring the proper amino acid, peptide bond it, shift it down. The used tRNA is gone. The A tRNA goes to the P site, a new tRNA comes into the A site, and it does it again. There's some really good animations I'm going to post for this, There's lots of good animations, but there's some I'll put like super cool, or make sure you watch this one for both the DNA replication, transcription, and translation. They're PBS animated, showing the actual molecular, in not like cartoony pictures like these, but actual kind of modeling of the molecules. And what's so cool about it is it shows it in real time, like how fast is this process actually happening inside of your cells right now. It's so cool. Okay, so this is the process of elongation. It's just going to continue down the line until we've got to have a stop point somewhere, right? So one of those 3 stop codons, when that shows up as the ribosome's just plugging along, adding to the polypeptide chain, once a stop codon comes into the A site, there's another kind of protein that comes in as what's called the release factor. So when the release factor comes in, it triggers our new polypeptide chain to be released. It's done. It's in that primary sequence. It's just a big long chain of amino acids that's going to start doing secondary and tertiary folding. After that release factor comes in and binds to the A site, it triggers everything to just kind of come apart. The small subunit separates from the large subunit. The tRNA goes back to being recycled. The release factor just hangs out in the cytoplasm. The 5' cap and the poly A tail have been removed. So now our mRNA is ready to be recycled. It does not get translated again in eukaryotic cells. So there's bacteria do something special I'll talk about in another slide. But now this whole mRNA is going to get recycled. All the individual nucleotides will be broken apart. They'll be shuttled back to the nucleus to be used again in another RNA molecule. It could be another mRNA or part of a tRNA or an rRNA. So it's just recycling those RNA nucleotides. And then the ribosome can go back and find another newly processed mature mRNA and make another protein. So it doesn't matter. They're the workers, right? So these are the production table. They will make whatever the instructions tell them to make. So they can make an enzyme, they can make a structural protein, they can make a membrane protein, they can make an antibody. Whatever the mRNA tells them to do, they just make whatever those codons tell them to do. They're not special. I mean, they're special, but they're not protein specific. They can make any protein that the mRNA tells them to make. Okay, so here is, this is a picture kind of putting it all together. This is not from your textbook. It's from an older text. You can see all the way from 2008. But I keep it in my presentations because I kind of like it. It shows the process of the master blueprints keeping in the nucleus. It shows the process of transcription happening off of the DNA. The RNA processing, removing the introns, keeping the exons. We have the 5' cap and the poly A tail as our little squiggly ribbon of mRNA comes out into the cytoplasm. In this case, the RNA or the tRNAs are kind of those bluish green tubes. But we have the binding, the coming together of the ribosomes onto your mRNA. We have our E site, P site, and A site, our growing polypeptide chain. And here's our amino acid tRNA synthetase charging up the blank tRNAs, and they will match the anticodon with the proper amino acid, so it matches the instructions. All right, so this is it. That kind of wraps up transcription and translation, that idea that the information coded for in your DNA is read into RNA, which is then translated into a protein. And that's how our cells work. It's so cool. Going all the way back to chapter 2 and 3 stuff for what proteins are, bonding and cellular structure with the nucleus and the ribosomes. It's just so cool how it puts all the pieces together. All right, so I think I got a couple more mini lessons to kind of wrap up chapter 13. So I will see you next time.